Hi, my name is Tyus Williams. I am a PhD student at University of California, Berkeley. My thesis advisors are Dr. Christopher J. Shell and Dr. Justin Brashares. And I'm doing my presentation on evaluating predator detection frequency in response to anthropogenic forces on East Bay shorelines. So here's what you can expect throughout this presentation. Things are going to be a little bit different for this one because I'm still in the developmental stages of my project, especially for chapter one. So I have some information to share, but I don't have a lot of like more analytical and quantitative like results to kind of show. But I'm sure hopefully it'll be very exciting as I go throughout the process of this. So you can expect an introduction. I'll go through basically kind of how I got to this project, background and history on the development of it, some of the methods and the development of the project itself, some observations and examples that I've gathered from what I've seen so far out in the field, and generally, kind of a simplified conclusion and just the general next steps of like where I'm going to go from here. And so for introduction, my name is Tyus Williams. I use he, him pronouns, and I'm currently a second year PhD student at UC Berkeley in the Department of Environmental Science, Policy, and Management. I perform my research in carnivore ecology and spatial ecology. I'm fundamentally interested in understanding how animals, especially large carnivores, navigate through a rapidly changing world and how they compete against each other under stressful circumstances. And this can be on a local scale, or this can even be on an international scale. I did my undergraduate thesis on jaguars, and now I'm here in the Bay Area launching one of my first chapters, doing more local, regional based research, which is very exciting. And so, similar in the way paradigms in society shift over time from being contended or reformed, my perspective as a scientist has changed throughout my years of learning. I approach my research with a multifaceted approach to effectively disentangle the deeply inherent complexity of the natural world. If you look over to the right, here's a visual representation for how I think about my research. It's kind of a glimpse inside of my mind, if you will, from the frameworks of the foundation being wildlife ecology, which is then informed my understanding of spatial movement ecology, to predation dynamics, interspecific competition, human-wildlife conflict, which then coincides with environmental justice, giving me a multifaceted ecological perspective that there goes back into informing my wildlife ecology, skill set in discipline. And this is all really important because effective research requires an intersectional and interdisciplinary approach for efficient problem solving. So just for some background context, matriculating into UC Berkeley, I was approaching my research interested in studying abroad in Ruaha, Tanzania, actually, for my thesis chapters. I drafted and submitted my application for a National Science Foundation fellowship, specifically the National Science Foundation, the Graduate Research Fellowship Program, which I did get. So hooray for that. That was a big win for me my first year. And I submitted this application under the prospect of understanding how human land use potentially exacerbates intrakill predation or interspecific killings amongst African carnivores, and how it may inadvertently result in further human-wildlife conflict. I actually have a graphic image, a conceptual figure from my NSF proposal visualizing a potential scenario within my research question, and this basically just shows kind of like almost like a cost-benefit analysis situation where a predator has to kind of pick between two different types of consequences, which is, experiencing habitat loss, and then, potential intrakill predation being caused because of habitat loss. And then this animal is basically put in the situation having to pick between, A or B, competing with more animals in A, or potentially experiencing persecution in site B. And so I thought this image was kind of cool because it kind of gives you an idea of like what I was trying to accomplish with my research, potentially out in Africa. Maybe I'll get back to it one day. And so just background context as well is, so some interesting things to know are, growing research from insights provided by, Caitlin Gaynor, I believe in 2020, and Justine Smith in 2019, and Kirby Mills in 2020, highlight the increasing concern how human-dominated landscapes modify the behavior of wildlife across time and space, holding severe implications towards organic niche partitioning, that is, habitat selection and dietary overlap, as I highlight in this figure over here to the right. And one of the things is that Africa has some of the highest biodiversity of species within the order Carnivora. And it was because of this, I found myself fascinated with this entanglement of human-wildlife interactions. I also just have another conceptual figure over here detailing just an idea of like what I was trying to accomplish out in my research potentially in Tanzania. This idea of just how different types of dietary overlaps can create these situations where animals have to essentially engage in particular forms of competition with each other, and that can result in either symmetrical killings or asymmetrical killings. And Tim Caro has a incredible paper out from the 90s where he details mammalian interspecific competition, specifically amongst African carnivore guilds. And but more importantly, like, why do I share all this? Because we're not talking about African carnivores or African wildlife species. We're talking specifically about research that I'm doing here in the Bay Area, and that's important because ideas change in response to collaboration. These are many of my lab members, my beloved lab members. They're like family to me, and I love them very dearly. This is Chris over here to the right, as well, the individual who I'm here in the photo with, holding a beer in my hand after we're celebrating a long semester of hard work. And ideas change in response to collaboration. Entering the Shell Lab and the Brashears Lab for my PhD fundamentally changed my approach to the questions I asked, not only on an international setting, but with an urban context as well. This inspired me to ask more questions over urban ecology and how I can remedy issues through a solution-oriented approach using wildlife research. And this would lead me to the development of my first chapter right here at the Bay. And so developing chapter one. So how did I end up collaborating with East Bay Regional Parks for my first chapter? Well, it all started from a conversation between three scientists over a growing conservation concern at the Martin Luther King Regional Shoreline. These three individuals being me, and Tammy, and Dr. Sue Townsend. And we had a just wonderful conversation about just some issues that were, emerging from just the past and how we can potentially try to ameliorate and resolve them. And so some background history on the development of Chapter 1. So in 1988, one of the East Bay's most significant wetlands restoration projects was completed at the Martin Luther King Jr. Regional Shoreline, which restored tidal flow to 71 acres of tidal and seasonal wetlands that had been filled in the mid-1980s. This project resulted in the reestablishment of native plants and the return of many species of birds. Thousands of migrating birds have returned to the marsh annually to rest and feed. For some species such as avocets, terns, egrets, and the endangered California clapper rail, and burrowing owl to live at the marsh year-round. And what's wonderful is school and volunteer groups utilize the restored marsh for teaching opportunities and community projects. Students learn about wetland ecosystems. They venture into the marsh in canoes to explore wetlands and participate in a wide variety of monitoring and restoration efforts. And this is all really important because, with any restoration success story, bringing native plants and animals back to the area presents the opportunity for wildlife communities to reassemble. This means recovering bird species and other wildlife groups are potentially exposed to predation by native wildlife, especially invasive species where natural removal via predation and competition doesn't affect them equally. And unfortunately, authorized removal of certain animal groups presented some complications for park managers mitigating prior management applications. And this is where the inception of my first project began in collaboration with East Bay Regional Parks. It was, having an understanding of all this information, this being presented to me when I was talking to Sue and Tammy, and then this, further information being provided when it comes to like the management constraints. And I was thinking to myself, I was like, well, how can I try to solve this, or how can I collaborate to try to resolve and present some quantitative information on, hopefully presenting more effective conservation applications for the future. And so, initially the beginning of my research was launched in the interest of understanding how the presence of fence lines influenced the detection of free-roaming domestic cats across all 11 East Bay shorelines. However, I found that the fence grids for sensitive shoreline habitats were not present across all my site locations, meaning I had to adjust my methodological approach. So I decided to shift more towards anthropogenic characteristics such as residential development, impervious surface area, and other environmental variables to unravel my question. And this is just a figure that I created for my first chapter here to try to detail the idea of what I had when it came to fence barriers and how animals potentially bypass them through their physical abilities. And so just some more background context is just, Felix Medina in 2011 put out an incredible paper detailing the ecological impacts of invasive cats on island endangered vertebrates around the world. And one of the things that was fascinating about this paper is they highlight that prior to this publication, a global review for island impacts was never done. Furthermore, information regarding feral cat impact was primarily qualitative on mainland areas and large islands. This piqued my curiosity to start investigating the ecology of free-roaming domestic cats on a spatially explicit local scale to understand what is happening here specifically in the Bay Area. I decided later on to incorporate a broad sample of mammalian mesopredators to ensure that my hypothesis is more robust under the question of human-wildlife intersections. So generating research questions in journals. So like what are the questions here with my research? And generally it's how is the detection of mammalian predators influenced by anthropogenic factors throughout the East Bay regional shorelines? How do covariates such as road density, housing density, and residential proximity potentially explain this? Furthermore, with the understanding that human-dominated landscapes can hold severe implications for the ecology of wildlife species in their respective communities, what does this mean for regional shorelines in the future, right? How do the answers here inform the socio-ecological discourse likely to unfold here in the Bay? And so here is my study area. This is just from Google Earth. I made a map, and you can see that I have these markers across all of my study, my little site locations where I place cameras. And so for my methods in development, so in the beginning of April, I placed 2 to 4 cameras in each shoreline, and they were each distance about 500 to 700 meters apart from each other. And I used Bushnell units, and they all either contained a 32 or 16 gigabyte SD card. Cameras were checked every 2 weeks at the beginning of the field season. We then transitioned to checking on them at the end of the month since our camera locations are reliable. Wildlife images are then sorted in Adobe Lightroom and uploaded to Wildlife Insights for photo scoring. All mammalian species are included for scoring, but people, also known as just human images, are removed. And lastly, a CSV file will then be downloaded to use for analysis in R and Esri software. And then I'll run all the appropriate modeling that I have when it comes to those layers of covariates that I'm trying to understand correlations to when it comes to my research. So I actually have a table here that I created to just show just like the species that I've actually observed in some of my shorelines. All of the highlighted areas with the letter Y basically indicate that these have been confirmed in my sites. And what's interesting is that domestic cats have been recorded on camera in 10 out of 11 sites. That's 91% of my locations where they're being detected on my cameras. The only animal that has equal presence is the northern raccoon, but you would expect this generally from a generalist species. Furthermore, the reason why we don't have any detections in Rat Key is because both cameras were stolen, making it a difficult area to continue surveying it, which makes me wonder if like I was capable of actually getting actual data collected back before my cameras got stolen. If I would have gotten a 100% presence confirmation of domestic cats in all of my sample locations. So pretty cool information. Yeah, we have to see pumas, but maybe we still got images to go through. And we actually, we got a variety of animals, on our cameras, from northern raccoons, from Big Break Regional Shoreline, to skunks as well. I was really delighted to see river otters pop up on my camera. I was not expecting them. And these are also being found in Big Break. It's the only place where the otters have been found. And getting coyotes in Carquinez, which has also been just delightful to see. And as you can imagine, cats in many of these areas as well. But certainly showing up at different times when it comes to, as you can imagine, coyotes being around, which is definitely they have some particular avoidance strategies to try to avoid being the next meal of snack for these animals. And so, and we're also getting some interesting species interactions and avoidance responses have been observed too. So this is a photo that two photos that I have from two separate isolated events. If you look at the time lapse, it's a 9-minute difference apart from each other of a feral cat coming in. This is in Carquinez. This is at camera 24, I believe. And coming in, and then 90 minutes later, a raccoon's coming back down the path, and you can see the feral cat is actually trying to avoid it. And so it just makes it's cool to see actual confirmed physical evidence of actual avoidance between animals, and the fact that maybe this feral cat actually just really doesn't want to deal with a raccoon coming across him and potentially having to engage in whatever kind of squabble or quarrel that they might have when it comes to maybe territory or land tenure. And so we also just have some other photos too, just purely here for humor. This is a big break. It seems like a raccoon stumbled across this skunk and kind of got into it. I don't think he was sprayed, or it was sprayed. I'm not sure what the sex was, but certainly was startled. That's a pretty high jump for the raccoon to make the realization that it might have been in a dangerous situation. But I also just found this to be pretty humorous, considering the fact that the images were fairly close together, and yeah, they're pretty much separated events. And so just moving forward too, we also got a lot of cats. I was talking to Tammy about this. Tammy was asking me to update her on how things were going, and I was telling her that like we actually got a lot of cats. I was expecting to get a significant number of them. I'm actually not entirely sure how many individuals we have across all the shoreline sites, but I'm getting way more than I expected in terms of just frequency, from just Big Break to Martin Luther King shoreline to Bay Point, in as well as Point Pinole. Excuse me, we have yet to get them in. Oh yeah, we got them in Point Pinole, in Miller Knox. We got them in just about every site except Racky, which is a little concerning, but hopefully interested in understanding more about that with the quantitative analysis. I'll run further. And I also just want to go into a little bit more detail too when it comes to just like the images that we're collecting here, is because I want to point out, we're collecting some interesting images on free roaming cats in some interesting locations. And so these two photos are both from Carquinez. The top one being from Carquinez 23, and the bottom one being from Carquinez 24. But I really want to point out something particular here in terms of the image, is that like keep in mind that like the linear distance for Carquinez 24 is 541.93 meters away from the nearest building. But however, this only tells me how far it is in terms of linear length without any landscape resistance. This excluding slope, elevation, and land cover, which can influence the movement ecology of an animal. This makes me wonder how these individuals are getting all the way over here, because it's a really fundamentally fascinating question. It's like when I saw this on the camera, I was like, where did you come from? Like, it didn't make any sense to me. But hopefully further analysis in GIS software will visualize, the types of landscapes these animals move through. And so I find myself asking more questions as the project progresses, and it's been very satisfying moving through it all so far. But more importantly, we have some interesting photos from the shoreline sites. We have predation events on camera. That's right. These are, I think, 3 or 4 different sites. These are 3. Correct me, excuse me, I'm wrong. These are 3 different sites with 3 completely different events of predation captured on camera. And some of these are actually reoccurring individuals, which is actually like a little bit disturbing to think about. So we have confirmed predation events in 4 out of 11 shoreline sites with reoccurring events from the same individuals. This spells for a concerning circumstance because we don't know the frequency for how often this occurs, nor the proportion/concentration of their diets consisting of native wildlife. This is something I hope to tackle in my second chapter, looking more closely at free roaming cat movement ecology via satellite collaring in diet concentrations using stable isotope analysis. That is a duck in that cat's mouth, which is a little sad, but cats are predators to a lot of native wildlife. So here's some additional thoughts that I have. A variety of animals have been observed across these shoreline sites, giving us a variation of diversity across the study area. This information will be used to understand the ramifications of anthropogenic factors on species diversity as well. And also, I'm also genuinely surprised by the abundance and frequency of domestic cat detections I've received on my cameras, primarily because there's a significant distance between each shoreline site, which tells me these are not only unique individuals, but separated populations, which is interesting to know. There's a lot to learn and discover throughout the development of this project. And currently I'm in the data management stage of filtering and sorting the images I collected over the summer thus far. I'm looking forward to the continued progression of this project and moving into the data analysis phase in the future. I'm always learning, and I will continue asking questions about the ecological connections happening here in the Bay Area. Science is a never-ending journey of unlimited potential if you have the means to continue asking exciting questions. Thank you for watching. If you have any questions, please let me know. Also, major thanks to Tammy, Lim, Dr. Sue Townsend, David Reinsch, Doug Bell, and all the staff at East Bay Parks for the assistance they've provided throughout the project so far. I also just want to note that this project is made possible under funding provided through the National Science Foundation, Graduate Research Fellowship, and permit authorization from the East Bay Regional Parks District. Thank you so much for all of your help, and yeah.